Hello. This presentation is entitled Technology and Bacteria, an experiment in DNA extraction, PCR, and DNA sequencing. It's presented by myself, Lindsey. On June 29, 2007, Apple released the first edition of the iPhone. Almost 13 years later, on September 20th, 2019, Apple released the 11th edition of the iPhone. And in those 13 years, the iPhone revolutionized not just the way that we communicate, but also the way that we travel and even the way that we learn. Regardless of what type of mobile phone you use, we use our mobile phones a lot. And with that comes a high likelihood of bacterial transmission between the surface the phone is resting on, the phone itself, our hands, and vice versa. A University of Arizona study found that the surface of a mobile phone is 10 times dirtier than that of a toilet seat. And in a study conducted by the National Institute of Infectious Diseases, it was found that high school students' mobile phones have more than 17,000 bacterial gene copies on their surface. What does that have to do with DNA extraction, PCR, and sequencing, you might ask? Well, let's understand what those are and how they work together. DNA extraction is simply breaking cells open and bringing the genetic material out of solution. Running a polymerase chain reaction is amplifying that genetic material so it can be run through gel electrophoresis and ultimately sequenced to determine what specific sequence it is. And what does that provide us with? Well, identification. It allows us to know what bacteria we're working with. And if we could find out what kind of bacteria were most commonly growing on the surface of our mobile phones, we might be able to understand how to kill that bacteria more effectively, or better yet, prevent it from growing in the first place. A possible bacteria we might find on the surface of our mobile phone would be a staph bacteria. Staph is found on our fingertips and on the surface of our skin, so it makes sense to find it on the surface of our mobile phone. Staph is a gram-positive, spherical bacteria. It's also a facultative anaerobe, meaning that it makes ATP by aerobic respiration if oxygen is present, but it's also capable of switching to fermentation or anaerobic respiration if oxygen is absent. And while we didn't take steps in this experiment to prevent the growth of bacteria on our mobile phone, we did attempt to identify what kinds of bacteria or what kind of bacteria might be growing on my personal mobile phone. I hypothesized that I would find a staph bacteria growing on the surface of my phone. The first step in verifying that hypothesis was to do a DNA extraction with the objective of isolating the DNA. So I swabbed the surface of my mobile phone, swabbed it into an agar plate, allowed that to grow in an incubator for a week, and then removed the colonies and placed them in an epitube. After adding the following buffering compounds and macromolecules to break open the cells and bring the genetic material into solution, I then incubated for an hour, centrifuged, and separated that solution from other particles that might have been found. Then it was time to bring the genetic material back out of solution by adding ethanol and ammonium acetate. I then placed it in an ice bath for 30 minutes, centrifuged and decanted the liquid away, and we were left with a pellet of genetic material in the bottom of our epitube. We then added 50 microliters of buffer for preservation before PCR. We then conducted our PCR with the objective of replicating and amplifying our DNA. After making sure that we had enough genetic material to work with, we created a master mix with 3 times the amount of ingredients that are listed. We then created a positive control and negative control and ran our PCR. I wanted to highlight specifically the Taq polymerase and the DNTP that were included in our master mix, as they were 2 of our most important aspects. The Taq polymerase was necessary because we were attempting to amplify or replicate DNA. And an important part of DNA replication, a necessary part of DNA replication, is having a polymerase. Taq is a great candidate because it's a thermostable DNA polymerase, which can also work at higher temperatures. The DNTP, nucleoside triphosphate, gives us the phosphates we need to form our phosphodiester bonds. We then ran our gel electrophoresis with the objective of separating our DNA into identifiable bands. We created a positive and a negative control, loaded our chambers with TAE along with our samples, and allowed it to run for 30 minutes or until it was about halfway down our apparatus. We then imaged our gel and proceeded with sequencing. Unfortunately, we did not have any identifiable DNA bands on our gel, so we had to work with, for our sequencing, a given unknown. With that unknown, we called out our bases in MegaX up to 300 bases and created 20 alternative sequencings using a randomizing process in Excel. We then ran a phylogenetic analysis in MegaX using the bootstrap method up to 500 for phylogenetic trees. We then did a blast search using the sequences that had a bootstrap number of 10 or higher to find the most similar sequences to the ones that we were given. These were our results. For our electrophoresis, we had nothing to view as no bands were visible. For our phylogenetic tree, we had to use, again, an unknown DNA to sequence, which is listed there on the screen. And our sequences that were above 10 bootstraps were 20, 5, 6, 16, and 10. And the blast search that was resulting from the sequences that we placed into blast search is listed there. Unfortunately, we really weren't able to verify our hypothesis because we weren't able to really extract the DNA successfully from our sample. So our result was inconclusive. However, what we learned in this experiment was an excellent way to be able to not only extract our DNA, but also sequence it if we can get a successful extraction. So what would be exciting to do is conduct this experiment again to be able to extract our DNA more successfully and then sequence it to be able to determine what kind of bacteria was growing on the surface of the iPhone.